my name is Connie. I'm an undergraduate at MIT, and today we're going to be doing a little bit more additional material on viruses. So previously in the viruses lecture, we were talking about phages in particular. In phages, the phages we were talking about were bacteriophages, which means the viruses that we were detailing specifically attack only bacteria. In this case, we're going to be talking about classes of viruses that carry RNA genomes. And for the most part, these types of viruses only attack plant cells and animal cells. And they attack animal cells more so than they attack plant cells. So we're going to start out with a few different classes of RNA genome viruses. So first off, we'll be talking about class 4. Class 4 viruses directly inject their RNA, and then these can directly be used as mRNA within the cells, and they can start translating viral protein immediately after infection. So this one has an immediate effect. In contrast, for class 5 viruses, these are going to be viruses that have an RNA genome that serve as a template for mRNA synthesis. So the RNA will go into the cell, and then using the replication or transcription parts within the cell already, they're able to make an mRNA strand out of that. So this one that it inserts is complementary to the one that it actually wants. And then using this two-step process, the viral DNA will be able to get mRNA that will be able to create viral proteins. Now for class 6 retroviruses, these are going to be very, very important because these are the type of viruses that make up HIV. And the disease form of that is more commonly known as AIDS. So retroviruses are responsible for AIDS. So the way that retroviruses works is that the viruses are equipped with an enzyme called reverse transcriptase. So when they inject their RNA, they also inject this very specific virus enzyme as well, called reverse transcriptase. Reverse transcriptase's job is to transcribe the RNA that it's injecting into DNA. And this is going to be double-stranded DNA. And then this DNA can then incorporate into the DNA of the host cell and be able to incorporate itself and then replicate, which is why HIV is more often than not in the lysogenic stage. And in that case, it's really difficult to detect when it is just in your body and not really causing any harm besides replicating cells that hold its DNA within it. And then that's why you have such an extreme reaction when the HIV does become active. And then that's the result of that is AIDS. So those are the different types of RNA viruses available. And as you can see, they can be pretty detrimental. So this is all important things to remember from what viruses are capable of and the many different types of viruses that are present in the world.